So in this video we're going to look at autosomal linkage, which is when you're going to be looking at two traits. So initially this might appear to be very similar to those dihybrid problems we've been working on. And it is actually quite similar in that you can have two traits, for example hair colour and dimples, so controlled by completely different genes. But the main difference here is that these genes are both found on the same chromosome. So we've got a worked example here to go through, and then we've got a question a bit later on. Now, we've got red hair and dimples found on the same chromosome, and red hair is recessive. So let's come up with some notation for this. I'm going to choose lowercase r to represent the alleles for red hair. So if you had that genotype, you would have red hair. Now, dimples are dominant, so let's go with capital D for dimples, which means that they will have dimples, but they will have dimples as well. Now, that means that in order to have no dimples, you need to be homozygous recessive. So that means that that is the genotype for no dimples. Now, we're not looking at these traits in isolation, we're looking at them together. So it says, work out the probability of two parents heterozygous for both traits having a red-haired dimpled child. So let's write out the cross first of all. We know that they're heterozygous for both traits, so they're going to have, well, red hair is recessive, so they're not going to have red hair. So let's go non-red and heterozygous, that puts them here, so they are going to have dimples. And this is the cross, non-red-haired dimples. Let's represent that with our notation here. So heterozygous for both traits, RR, D, D, and RR, D, D. Okay, this is so far is exactly the same as the dihybrid, but this is where it gets different because it says the dominant alleles are found on the same chromosome. What does that mean? Let's draw a cell from this individual, and rather than having R and D on separate chromosomes, these are going to be on the same chromosome. Now they told us that the dominant alleles, that's both the dominant R and the dominant D, are on the same chromosome. That means that the lowercase ones, the recessive alleles, must be on the homologous chromosome. And that's going to be the same in this one over here. This means that when these cells undergo meiosis, instead of getting the usual variety of gametes that you get from a dihybrid cross, we're going to be a bit limited. Normally, with two traits, you would be able to make 4 different gametes. You need to have an R and a D in each one. So normally what you would do is you say, okay, well we could have that, we could have that, we could have that, and we could have that. But now, if you look, because the capital R and the capital D are physically attached, when we pull these chromosomes apart, we can make this gamete, but there's no way we can make that one or that one. We can, however, make this one. So we've effectively limited the variety of gametes that we're going to produce. So when you come to do your Punnett square, it's actually a lot simpler than it was for the dihybrid. You've only got 2 types of gametes from each parent. Okay. There's our first offspring. They will have both dominant conditions, so they will have dimples and they will not have red hair. This individual is going to be heterozygous for both traits, but again, they're going to be non-red-haired and they're going to have dimples, so just the same as the parents. The same is true of this one here, but it's this one down here. This one is going to have red hair and no dimples. Okay. It says work out the probability of 2 parents having a red-haired dimpled child. Well, are any of these having red hair and dimples? That's non-red, that's non-red, that's non-red. The probability of having a red-haired dimpled child is 0 because it's simply impossible from this combination with linkage. Okay, because they're on the same chromosome, you're never going to get those 2 conditions. You're never going to get a red-haired dimpled child. There we go. It is impossible. Now, in actuality, what you sometimes find when you do this cross for real is that it is possible in very rare instances, to get these kind of combinations that we're being told are impossible. We can't make them. And that's because when you actually watch meiosis occur, in a small proportion of cases, it doesn't just happen where you separate these out nice and neatly and have those two traits going to one gamete and those two traits going to another. You can, of course, get crossing over occurring. So if I show you that by imagining that these two cross over during meiosis at this point, and what they will actually do is that they would swap bits of the genetic material from this chromosome onto this one and vice versa. And what that will do is it will let you create this gamete and this gamete, but in very, very small numbers. Now, we don't see any of that in our Punnett square over here. The only time that you'll ever find that is an issue in a question is when they show you the frequency of offspring. And if you get any offspring that are occurring and they're at a very, very low ratio or low proportion, it's very likely to be due to crossing over. We're talking about maybe 1 or 2% of the offspring. That's often due to crossing over, and that's going to feature in our next worked example. So now you've got the hang of this. Here is another problem. You can pause the video at this point and you can practice working through this part 1 of it. But just be advised that over the page you've got this bit, which this is linked to the same question, and this is the one which is actually giving us the frequency of offspring. So look, we've got these two phenotypes that are getting most of the offspring, but these ones here are cropping up with about 5% frequency. There's very few individuals that are cropping up with these conditions. That's to do with linkage, and that's to do with crossing over. So this is a double-sided question. So pause it now, and I'm going to work through it. Okay. So, just as before, it's an autosomal linkage problem with dihybrid, but with linkage. And we've got two different traits. Colour of Drosophila, they're flies, black and brown. Black is dominant. And we've got wing shape. Normal wings are capital N, they're dominant, and vestigial are lowercase n. Okay, so you're going to be writing genotypes out like this. Couple of B's, couple of N's, and variations thereof. Okay. So this would be a black individual with normal wings. But let's have a little look at what we are doing. The loci for these two genes are on the same non-sex chromosome. B and N are linked. Draw that to remind myself. And B and N are linked. Okay, that means they're on the same chromosome. And this is important because this is going to affect which gametes we can make. We start with our first cross here. A pure breeding, that means homozygous, black female with vestigial wings was crossed with a pure breeding brown male with normal wings. Okay, let's have a look at that. Pure breeding black female, so that's black, and with vestigial wings, so she's going to be that. She's crossed with a brown male with normal wings. Is pure breeding for both traits again. Work out the genotype of the F1 generation. Well, this is actually nice and easy. So, look, if we're doing this cross, little n's times B, N. Regardless of linkage or any of that kind of thing, because these are homozygous, they can only produce one gamete each. That's the gamete over there. That's the gamete over there. Fuse those together. I don't even need to do a Punnett square. We know we're going to get this. Okay? Heterozygous for both traits. That's the F1 generation right there. 100% of them are going to have that, and they are therefore going to be black body, and they're going to have normal wings. Now we're going to take one of these, an F1 female, and we're going to cross it with a true breeding brown vestigial winged male. Brown and vestigial wing, they're the two recessive traits, aren't they? So brown vestigial wing is going to be that. Now it says next, 1,600 offspring were produced. Work out the phenotypes and calculate the expected number of offspring of each type. So this is our dihybrid cross, which means that I've got the phenotypes up there. I've now done the parental genotypes here. So let's work out the gametes. Now normally, with a heterozygous for both traits here, you'd go into it and you'd start working out there are 4 possible gamete combinations. But remember, linkage. So I can actually only make that gamete and that gamete by meiosis. And this one can only produce one type of gamete. So my Punnett square, really, really easy. So we're going to have this possible genotype of the offspring, and we're going to have this possible genotype of the offspring. Let's have a little look at what those are. So we're going to have a black, normal, winged individual, and we're going to have a brown, vestigial winged individual. And if you look at it, the ratio is 1 to 1, isn't it? 1 to 1. And they've told us that 1,600 offspring were produced, so we would expect 800 of each of those types. Let's look over the page at the second part, and you'll see that the actual values are, well, they're pretty close. We're expecting black normal, 800. These are our expected values. And we're expecting brown vestigial, 800. And we're not far off. But look, we've got these ones creeping in. These impossible combinations. And my advice for that, when you get these ones, figure out what the genotype has to be. Black vestigial wing. Now, if they're vestigial, then they have to be that one. And if they're black, then they need at least one of those. And it doesn't actually matter what the next allele is. Now, brown, they've got to be that. And normal wing, they need at least one of those. So let's have a little think about the parental genotype and the parental gametes, and then we'll figure out what kind of crossing over has occurred. Now remember, for your parents, for this second cross here, one of them is homozygous for both traits, so it's only going to produce this gamete here. So we know that each of the offspring must have had a BN gamete. So we know that there must be a lowercase n from each of them. And we know that there must be a lowercase b. So we've now figured out the genotype of both of these ones over here. But how on earth do we get this combination of alleles? Let's have a look at the crossing over that must have taken place. So I'm going to put my homologous chromosomes next to each other. I'm going to put the B's at the top and put the N's at the bottom, so that we know that during meiosis we would normally be making that, and we would be making that. But actually what we've established, if this allele and this allele came from that gamete, we must have had a lowercase b capital N. So we need a lowercase b capital N. And actually if we have those ones were from this gamete, we must have had a capital B lowercase n. We must have produced gametes in these arrangements with these alleles in. The only way that that is possible at all is if these have crossed over at this point here and those B alleles have been swapped over. If you think about what that would give you, it would give you, capital N is where we left it, it's now got a lowercase b at the top, and then this one here, the N is where we've left it, but it's now got a capital B at the top. And look, that will lead to these gametes at the bottom. It's happened in very low proportions, it's quite unlikely, but that is how you end up with these very rare phenotypes. Okay, so that's autosomal linkage with crossing over. And the key to look for in a question stem that tells you you're going to need to think about that is the word linked. They are linked, or they might sometimes just be really clear and say they are on the same chromosome. Okay? I hope that was helpful.